Proper timing of meals can enhance exercise capacity and aid in the recovery and tissue repair following exercise. Consuming a carbohydrate-containing meal 4 to 6 hours before exercise ensures adequate reserves of glycogen, or stored carbohydrate energy, in the muscle and the liver. An additional carbohydrate plus a protein snack consumed 30 to 60 minutes prior to exercise may protect against energy depletion towards the end of an intense exercise session, as well as help to prevent the breakdown of protein in the muscle tissue. During prolonged intense exercise, greater than 60 minutes, beverages containing carbohydrates and electrolytes should be ingested every 15 to 20 minutes to prevent low blood sugar. Post-exercise nutrition is important to help to replenish glycogen stores and to repair muscle tissue damage during exercise. The International Society of Sports Nutrition recommends protein and carbohydrate consumption within 3 hours after exercise. Elderly individuals may require greater post-exercise protein intake to maximize the recovery. One study showed 20 grams of post-exercise supplemental protein maximally stimulated muscle protein synthesis in young men. Another study found in elderly men 40 grams of post-exercise whey protein enhanced muscle protein synthesis more than 20 grams of whey protein. Studies suggest caffeine ingested before or during exercise enhances endurance exercise performance. Emerging research also suggests caffeine aids in short-term high-intensity burst activity performance. For example, competitively trained males who ingested 5 mg per kg of body weight of caffeine lifted more total weight on the chest press, and they generated greater anaerobic power. This dose of caffeine corresponds to about 2 to 4 cups of coffee for a 170-pound individual. Approximately 150 to 300 milligrams of caffeine, or about 1 to 3 cups of coffee, has been shown to improve concentration and decision-making during and after exhausting exercise. Possible mechanisms for the ergogenic effect of caffeine include increased fat burning, reduced fatigue, central nervous system stimulation, and reduced perception of pain. The stimulating effect of caffeine is primarily due to its ability to block adenosine receptors in the brain. The possible side effects of caffeine consumption include an increased heart rate, disturbed sleep, and nervousness. These are generally less pronounced with lower doses. The response to caffeine varies considerably from person to person. A recent review suggested older adults may be more susceptible to the sleep-disrupting effects of caffeine than younger individuals, so awareness of sleep quality and modifying caffeine use appropriately are important. Creatine is a compound and it's naturally produced in the body, and it can also be obtained through the diet, predominantly through meat and fish. Not only is supplemental creatine one of the most popular and well-researched ergogenic or performance-enhancing aids used by athletes, it's also an effective agent for preventing and slowing age-related muscle loss, known as sarcopenia, and it has improved cognitive performance in the elderly. Mouse studies indicate creatine may hold potential anti-aging effects. Numerous studies have shown creatine supplementation can enhance muscle mass and enhance athletic performance. Creatine is most effective as an aid to high-intensity, short-duration activity, like sprinting or weightlifting, which derive energy from creatine phosphate. In older adults, creatine supplementation, with or without resistance exercise, has enhanced muscle strength and mass. It's increased bone strength and slowed the rate of sarcopenia. Furthermore, according to one analysis, combining creatine supplementation with muscle strengthening exercises is more effective than exercise alone in increasing muscle mass, strength, and functional performance in older men and women. Creatine doses used in studies that enrolled aging subjects typically ranged from 5 to 21 grams per day for a 150-pound individual for limited periods of time. Taking creatine supplements with carbohydrates or protein and carbohydrates can increase the creatine's muscle retention. L-Carnitine is a compound obtained from food and is synthesized in the body from the essential amino acids lysine and methionine. It's required for the burning of fat for energy production within the mitochondria, and it can act as a free radical scavenger. Studies have demonstrated that L-Carnitine supplementation can improve exercise performance and recovery. In a randomized, double-blind, placebo-controlled trial, healthy male volunteers who ingested 2 grams of L-Carnitine along with 80 grams of carbohydrate twice a day for 24 weeks exhibited a 21% increase in muscle carnitine content, compared with no change in the control group. This was associated with reduced perception of effort and improvement in exercise performance. By reducing free radical generation and muscle soreness, L-Carnitine supplementation supports muscle recovery after strenuous exercise. In a placebo-controlled trial in healthy young men, oral supplementation with 2 grams of L-Carnitine for 2 weeks resulted in significantly reduced markers of oxidative stress and muscle damage following an acute bout of exercise. The essential branched-chain amino acids, leucine, isoleucine, and valine, are important for the synthesis of muscle protein, and they're burned by muscle cells for energy. Human and animal studies have shown that supplemental intake of branched-chain amino acids increases exercise endurance. In a double-blind, placebo-controlled study, branched-chain amino acid supplementation for 3 days increased fatigue resistance and enhanced fat burning for fuel during an exhaustive bout of endurance exercise that caused glycogen or stored carbohydrate depletion. Like other essential amino acids, branched-chain amino acids function as precursors or building blocks for muscle protein synthesis. Importantly, branched-chain amino acids, especially leucine, also exert anabolic effects by directly stimulating muscle growth and by inhibiting muscle protein degradation. By reducing the breakdown of muscle proteins and promoting protein synthesis, branched-chain amino acids improve exercise recovery. In a study in long-distance runners undergoing intense training, branched-chain amino acid supplementation reduced the soreness and the fatigue, as well as markers of inflammation and muscle damage. Vitamin D plays an essential role in bone metabolism, muscle function, and immune health. Sufficient blood levels of vitamin D are important for musculoskeletal injury prevention and recovery, and they're associated with reduced inflammation and pain, stronger muscles, and better athletic performance. Apart from its role in preventing fractures and muscle injuries, research also suggests vitamin D can have performance-enhancing effects. Unfortunately, many athletes are vitamin D deficient. Trials of supplemental vitamin D at doses of 3,300 to 5,000 international units per day have found improvements in sprinting and jumping performance, as well as increased circulating testosterone. One team of scientists suggested supplementing with 4,000 to 5,000 international units per day of vitamin D3, along with 50 to 1,000 micrograms per day of a mixture of vitamin K1 and K2 to complement vitamin D's role in the bone and the calcium metabolism. This could support athletic performance by improving recovery time and muscle function. Glutamine, because it's synthesized in the body, is a non-essential amino acid. However, glutamine becomes conditionally essential when blood levels are reduced in times of illness or stress. Glutamine plays a role in immune response to muscle damage. In a controlled 2-week trial in male college-aged martial arts athletes, supplementing with 3 grams of glutamine daily for 2 weeks reduced the muscle damage, and it prevented immune function decline, including during a strenuous training period. A controlled clinical trial that used 10 grams of glutamine daily for 3 weeks in athletes undergoing intensive training found an improvement in immunity, as was evidenced by white blood cell profiles, including an increase in natural killer cell activity. Another controlled clinical trial found athletes who were given 5 grams of glutamine immediately after and 2 hours after intense prolonged exercise reported roughly 40% fewer upper respiratory infections than those who were given a placebo. Produced by the adrenal glands, DHEA or dihydroepiandrosterone, along with its sulfated form, DHEA-S, is the most abundant steroid hormone in circulation. DHEA is the precursor of sex hormones like estrogen and androgens. DHEA levels peak around the age of 25 and decline by roughly 80% by the age of 75. Studies show DHEA supplementation has exercise-enhancing effects. In a study in elderly men and women, DHEA supplementation significantly enhanced muscle growth and strength in response to resistance exercise. In a randomized control trial, a single dose of 50 mg of DHEA increased free testosterone levels above the baseline in middle-aged men. This dosing was followed by a bout of high-intensity interval training, after which free testosterone remained elevated in the DHEA supplemented middle-aged individuals. Whey protein, a group of milk-derived proteins with a high concentration of essential amino acids and branched-chain amino acids, activates muscle protein synthesis and recovery in response to resistance exercise. Whey protein supplementation significantly decreases body weight and body fat, and it increases lean body mass when combined with resistance training. Whey protein is rapidly digested and absorbed. Leucine, one of the branched-chain amino acids, in which whey protein is especially rich, plays an important role in muscle protein metabolism, healthy glucose metabolism, and body weight maintenance. In one study, whey protein given to healthy subjects during recovery for maximal effort exercise significantly increased the amount of muscle satellite cells. These satellite cells, or stem cells, are essential for muscle regeneration. In another study, high leucine whey protein hydrolysate was more effective than a placebo at increasing the muscle and tendon growth after 12 weeks of leg resistance exercise. HMB is a metabolite of the amino acid leucine, and it helps to maintain muscle function and supports muscle growth and strength. HMB preserves the structure of the muscles, and it supports resistance and endurance training performance. One potential mechanism of action is the regulation of cell signaling pathways involved in protein synthesis. In a randomized, placebo-controlled, double-blind study, 19 healthy older adults were confined to bed rest for 10 days, and then they underwent 8 weeks of resistance training programs. The subjects took either a placebo or 1.5 grams of calcium HMB twice a day for 5 days before bed rest throughout the rehab program. Those who took the placebo experienced a significant decrease in lean body mass after being in bed rest, while nearly all of those in the treatment group had preserved the muscle mass. In another randomized placebo-controlled study, 13 subjects who were accustomed to intense endurance exercise received either a placebo or 3 grams of HMB per day. After 6 weeks of daily training and supplementation, all subjects went on a 20-kilometer or 12-mile run, and then were assessed for muscle damage. The post-run increases in the levels of creatine phosphokinase and lactate dehydrogenase, 2 markers of muscle damage, was reduced in those who were taking the HMB when compared to the placebo. In another double-blind, randomized, placebo-controlled trial, approximately 80 people over the age of 65 were divided into one of 4 groups. 2 non-exercise groups, one of which took a placebo and one that took 3 grams of HMB twice a day, and 2 resistance exercise groups, one of which took a placebo and another that took 3 grams of HMB twice a day. Resistance exercise improved the lean body mass and the performance measures like hand grip strength while supplementing with the HMB. The HMB in the exercise group showed improvements in weight loss and fat loss, and the authors concluded that strength, muscle quality, body composition, and functionality in healthy older men and women can be improved through taking HMB, with or without resistance training. In another trial, 20 men experienced in resistance training were randomly assigned to take 3 grams of HMB, the free acid form of HMB, or placebo before undergoing a resistance training session. Measurements of muscle damage, muscle protein breakdown, and subjective exercise recovery were then taken. The results suggest the HMB, when given to the trained athlete before exercise, can reduce muscle damage and subjective recovery times. A meta-analysis that examined 9 studies determined that HMB supplementation during resistance training supports overall and leg strength gains in previously untrained men. Carnosine is a substance naturally produced in the body from the precursors beta-alanine and histidine. It's highly concentrated in brain tissue and muscle, although well known for its anti-glycation effects. Accumulating evidence suggests carnosine plays important roles in exercise performance and skeletal muscle health. The carnosine component beta-alanine also displays ergogenic properties. As it's a precursor to carnosine, multiple studies have used beta-alanine supplementation to increase carnosine levels and to enhance exercise performance. Carnosine protects muscles from exercise-related oxidative stress, and it buffers lactic acid buildup to reduce muscle fatigue. A clinical study in 14 male athletes found supplementing with 4 grams of carnosine daily for 2 weeks led to a significant attenuation in exercise-induced glutathione loss, while also reducing markers of oxidative stress. A meta-analysis of 40 individual studies comprising 1,461 participants identified a significant positive overall effect of beta-alanine supplementation, ranging from 2 to 6.4 grams per day for 3 months, supporting the efficacy of increased muscle carnosine on improved exercise performance and capacity. A randomized placebo-controlled trial examining 23 highly trained judo athletes found participants who consumed 6.4 grams of beta-alanine per day had a significant improvement in exercise endurance at 4 weeks. In another controlled trial, 30 healthy, strength-trained individuals were assigned to a placebo or 6.4 grams of beta-alanine daily for 5 weeks, after which the beta-alanine group showed significant improvements in maximum strength and power output. A study that enrolled 20 healthy participants found supplementing with 2 grams of carnosine and 2 grams of beta-alanine 4 hours before exercise tests improved some of the measures of muscle performance and endurance. In 2 double-blind placebo-controlled crossover studies, supplementing with 20 mg per kg body weight of carnosine plus a methylated version of carnosine with improved half-life led to significantly higher power on a standardized fitness test following 6 minutes of high-intensity cycling. In another randomized controlled trial of 50 people with stable chronic heart failure and severe left ventricular systolic dysfunction, already on optimal medical therapy, 500 mg of carnosine daily in lozenge form for 6 months led to improved walking distance in the 6-minute walking test and increased aerobic capacity during exercise. Overall, many studies have shown positive effects of supplementing with carnosine and or beta-alanine for exercise and sports performance. However, the literature is not completely consistent. Several studies have reported little or no effect from beta-alanine supplementation on measures of exercise performance. Some authors have suggested the variability of the observed effects might be due to variable physiological demands of different sport activities on baseline characteristics, such as overall nutrition, degree of fitness, and sleep habits. More studies with large sample sizes and rigorous methodologies would be needed to clarify the efficacy of carnosine and beta-alanine on exercise performance. For more information on herbs, supplements, and natural treatment plans, check out my website.